Alright, so I wanted to share this. This is my favorite patent. The title of the patent is Phytocannabinoids in the Treatment of Cancer. So we're talking about plant-based, plant-derived cannabinoids, not synthetic cannabinoids put together in a lab, and not cannabinoids that are found in our body like anandamide and 2-AG. So these are exogenous, plant-derived cannabis compounds. We're talking about THC, CBD, CBN, CBA, or no, CBC, a bunch of other ones. So this is the abstract, and this is really all you need to know. All the rest of it is a bunch of medical jargon. But this invention, the invention that is going to be owned by GW Pharmaceuticals, relates to the use of phytocannabinoids either in an isolated form or in the form of a botanical drug substance, a BDS, as a prophylactic or in the treatment of cancer. Typically, the cancer to be treated is cancer of the prostate, breast, skin, glioma, colon, lung, or bone or lymph metastasis. These phytocannabinoids may be used in combination with other cancer treatments. So I'm going to run through this real quick. This invention relates to the use of plant-based cannabinoids, so marijuana constituents, in an isolated form, like they just take out the THC in an isolated form from the phytocannabinoids, so from the plant still, or in a botanical drug substance, which is the whole plant extract, much like you would find a lavender essential oil. It's the same kind of thing. You can cold press it, you can dry sift it, you can um, what is it called? You can use ice with it. You can use CO2, which is what we're going to get into here in a second. And so the BDS is just a botanical drug substance taken from the whole plant or the whole flower. So they can be used as a prophylactic, so a preventative measure, or in the treatment of cancer. Then they give this list of cancers that can be used or be treated by cannabinoid-based therapy. The first one, prostate, we have found that whenever the prostate, which has CB1 receptors on it anyway, but whenever it turns cancerous, there's a higher expression of CB1 receptors on the outer surface of the tumors. And so it's just an indication that the body is aware that there is a tumor growing. And so it expresses these CB1 receptors and probably floods the bloodstream with anandamide to go and attach to those receptors to keep the cells from proliferating and the tumor from growing overall. It gives a good list, and in the list, of course, along with prostata's breast, there's 3 subtypes of breast cancer. I was watching a video just last night of a presentation from Christina Sanchez, who's behind a lot of the research behind this group as well, because a lot of those inventors, the ones that came out of Spain, work in a clinic at a university, or in a lab at a university, and she is part of that group in the lab. She has done studies with all 3 types of subsets of breast cancer, and they have found the botanical drug substance is more effective than an isolated THC molecule, and that it works exactly the same whenever they've used it. On a graph, she showed the graphs, and the standard chemotherapy for breast cancer, I can't remember the name of it, but the function of that, or the potential of that, is exactly the same as the BDS, the botanical drug substance. So it's has the THC, CBD, and it's full of terpenes as well. They did a side-by-side test as well as just terpenes and cannabinoids, or just terpenes alone. The terpenes, which are the smell compounds, did not have any action against the breast cancer cells, but we know that the cannabinoids did, equal to the standard chemotherapy that is given to breast cancer patients. So if that's the case, by using that in conjunction with cannabinoids, you can use less chemotherapy, if we even have to go to that point. You can use less chemotherapy or less radiation or a smaller incision for surgery in order to get what is there out as far as the cancer. Now, what I love about this one is it actually shows you how to do what they're saying. And there goes all my stuff. I need to go up here and figure out where I'm at now. Sorry about that. Happens all the time. I need to watch my finger as it keeps moving. As you can see, there's a lot of information here concerning cannabis and cancer. And there we are. So, this graph right here is an overview of the botanical drug substance preparation. This is kind of whenever you talk about FICO or Rick Simpson oil or any kind of extracted oil that is crude that we make, as a population, apart from being prepared in a lab and all this stuff. And you're going to see how they prepare it. This is GW Pharmaceuticals. They're the ones who own Epidiolex. That Texas and the Teacup program already purchases their products, Epidiolex. And so the relationship that GW has with the state of Texas ought to be enough to show that Texas has at least accepted the therapeutic value of the products coming out of GW, namely Epidiolex, which is I think 100 milligrams of THC per milliliter. So anyway, they're describing their process for getting the BDS, which is what is killing all these cancer cells. Starting out, of course, you're going to grow the plants. Then you're going to harvest the plants after they've grown and produce flowers. You're going to dry and remove the stems. You're going to mill it up. And so now I'm going to go over this one more time. So what Rick Simpson does is he's taken plants that have already been grown, already been harvested, and already been dried. And some of them still have stems. In the video, you'll see that he doesn't mill it. He puts it into a 5-gallon bucket and uses a stick to crush it up. So that's really the only difference between Rick Simpson, his method, and then this one right here. This is where it definitely changes up. So they decarboxylate it separately in like an oven type format. Simpson, it decarboxylates as the alcohol is being evaporated off of the mixture, the BDS mixture. So they don't use alcohol, they use liquid CO2. Whenever you take CO2, put it in a subcritical manner, use it in a subcritical manner, it goes liquid. You can use that liquid to wash the cannabinoids off of the foliage from the plant matter, just like the alcohol does, or we always do food grade alcohol, so Everclear is what people suggest, and so I'll do that as well, just suggest some sort of Everclear food grade alcohol. And then they put it inside of a freezer, they winterize it, they filter off the waxes, and then they evaporate the solvent, and then that is their final product, the BDS. So again, this is exactly the same process, essentially, that it's pretty much as close as you can get to what Rick Simpson does. So any of the Rick Simpson oil, the cannabis extract, the FICO, anytime you hear of that or somebody tells you about it or you're telling somebody about it, they're basically making this. So the problem that I have with FICO is going to be if the person used pesticides, herbicides, fungicides. And so yeah, basically I just wanted to show you all this. You can take a screenshot of this first page, you can get the number, share this. This is not in the United States clearly, but the whole purpose of the scientific method is to have it be repeatable. And so this is repeatable. You can see it was done out of Italy and Japan and Great Britain and Spain. So these are all spread out and all their data points in the same direction. So it's called the coalescence of data, or the convergence of evidence, that these seemingly different researchers have all come to the same conclusion. And from that conclusion, from the evidence that's been amassed and put together, we can draw a pretty definitive reason to go into clinical therapy or clinical trials. Everyone that has been a part of this research, if you look up their own private research papers and publications that have been put in medical journals, medical literature, you're going to find that at the end of all their studies, they're all pushing for clinical trials. So that's why we're at the next step here with clinical trials. Okay, again, phytocannabinoids in the treatment of cancer. That means plant-based, plant-derived cannabinoids washed off of the flowers of the female plant are useful in the treatment of cancer. And they're using a BDS, a botanical drug substance, to do so. Alright, homies. Catch you later.